Oh, me again. So it's me again. And I'm here actually because, unfortunately, this year we moved the conference. The conference, the Focus on Lyme conference for the past 3 years has been in February. And this year we moved it to April. And as a result of that, the team from the Translational Genomics Institute, one is at the CDC, one is in Washington, D.C. And so I am here to give you a brief update on the progress that the team has made on the development of a direct kind of test for the presence of Borrelia burgdorferi. So, all right, so at this point, I think, can we agree, or at least potentially agree, that there are limitations to the existing diagnostic standard for Lyme disease? There are biological factors that need to be investigated, and I think finally are being investigated at this point. There are technological factors related to that test that cause limitations. And one of the, says this one. Just go back, nope. Okay, so what I want to point out is that the CDC explicitly, by explicitly I mean they specifically state these are the tests that you're supposed to use. They specifically recommend the two-tier serology as the diagnostic standard and go so far as to explicitly name tests that should not be used. And if you look down that list, it's basically every other test other than serology. And as new tests come out, they add to this list and say, we cannot recommend this test. And one of their reasons why is because these are not FDA cleared or approved types of tests, which, you know, when the CDC says, yeah, well, we don't want you using these tests, I think that in and of itself limits the tendency for people to try to go through that FDA approval process, which is hugely expensive and requires much resources. So the state of the testing is pretty bad at this point. And there has to be new things coming down the line. And the good news is there are. There's a number of technologies that are being used for other things, including what Garth just spoke about. There are technologies that could be applied to the diagnostic of Lyme disease, and that would include those that directly detect Borrelia burgdorferi or other species of Borrelia as the cause of Lyme disease. And then there are those that indirectly measure the response of the host, meaning us, to infection. And so all of these are promising and need to be carried up with. And I think after listening to the talk about what's going on with the immune system, I honestly believe that it needs to be a combination of both, a direct test to see if the presence of the microbe is there, and also an indirect way to determine what's happening with the person's immune system or other body systems. So with that in mind, I think the consensus has always been that it would be, for the sake of patients, it would be excellent to know if they still had the microorganism, if they still carried Borrelia burgdorferi even after treatment, or even before treatment, or if the bacteria was there. Because really, it comes down to whether or not to prescribe antibiotics for a disease that may or may not be a bacterial infection. And because antibiotics have side effects, it's always been a goal to say, is the bacteria still there or is it not? And so 2.5 years ago, Tammy approached the Translational Genomics Institute because that's what they do. They do cancer genomics, and so they go looking for unique signatures, and they also have a pathogen group that looks for different ways to diagnose bacterial infections that have routinely been very difficult to identify, such as tuberculosis, such as what's called here the valley fever, and they just recently succeeded in developing that test. And so the method that they use for those is called amplicon sequencing, and they developed an assay, or have been in the process of developing an assay that they called Lyme-seq, because the technology they're using is based on the Illumina sequencing platform, is using next-gen sequencing, and Lyme-seq just sounds pretty good, right? Copyrightable. So the method is a PCR type of test, but instead of going after one specific target, it actually is multiplexed, meaning it goes after many different types of targets. And, you know, at this point, it's not just Borrelia burgdorferi, so the test itself has over 30 different assays for several different Borrelia burgdorferi targets, but also other tick-borne microbes that are known to be co-infections, such as Ehrlichia, such as Anaplasma, and then also, other conditions that could also be misinterpreted as Lyme, and then also with an internal control, because obviously there's going to be some human participation in that. And so this is being, after the amplicons are identified, they sequence them using next-gen sequencing and can come up with whether or not there are 30 different things present based on if they have DNA or RNA in the blood. And because this is a blood test. And so the preliminary testing, once it was developed, the preliminary testing with spiked blood, which is not shown here, was very positive. By spiked, I mean that they had blood samples that they actually added the bacteria to at decreasing rates. Turned out that it was very good at detecting the pathogen itself. And then they also looked at how well the assay worked when it came to looking at pathogens in ticks themselves. So they got a series of Ixodes pacificus ticks, which are the type of Ixodes species that are common to California. So these were collected in California. And they had been previously tested at Northern Arizona University by Nate Nieto's team. And so when they compared the two results with LymeSeq and with the testing that he had done, they were able to identify all the same pathogens. As you can see, the green checks and the red X's indicate what they were able to find in those types of ticks. And then the next question was, does LymeSeq detect Borrelia in clinical samples? And so at about the same time all this was going on, there was an outbreak of a disease called relapsing fever in a camp in Arizona. And relapsing fever is caused by Borrelia, just not Burgdorferi. It's caused by, actually there's a number of causes of relapsing fever. And it's just not caused by Borrelia burgdorferi. And so they were able to acquire those samples because they were trying to figure out what the outbreak was on another scale. And so when they looked through the samples, they were able to find 8 out of 8 positives for the Borrelia hermsii bacteria, which you can see the results from that. And as a result of this study, which they reported on at a previous focus on Lyme, what they found was when it came to detecting DNA, they found 200 reads, but they found 200,000 reads for the RNA. So that, of course, indicated that the RNA, detecting the RNA from Borrelia without adolescent and nucleic acids, was going to be more likely to get a positive response than just using DNA alone. And the good news is LymeSeq looks for both DNA and RNA from the pathogens. And so then the last question to be answered was whether or not it would work on archived patient samples, meaning from a biorepository of samples that had been previously categorized and put into a biorepository. So those are the results of that first initial study. The biorepository was acquired through the Bay Area Lyme Foundation. They were characterized according to whether the patients presented with the so-called acute presentation of Lyme disease, which of course is the bullseye rash and positive serology. And they were blinded as to what was what when they got the test. And so what you can see in the first row, the first 10 were actually confirmed Lyme disease. So those are the people who were confirmed with Lyme as a result of having the bullseye rash and having positive serology. Negative controls, probable Lyme, which is the nonspecific symptom and some weird serology result. That's my interpretation of that, okay? And then the nonspecific symptom group, which they had no diagnosis. And when you look through that group, so for the PCR positive, the test results that were known to be positive for PCR, LymeSeq in that first group picked up all of them. All 3 of them were positive by LymeSeq as well. All of the negative controls were negative, which means that this is a very specific assay, and that's a good thing because that would mean no false positives. And then the really good thing is that if you look in those other 2 groups, there are 4 additional samples that were tested positive. These are people who would be told that they did not have Lyme disease if they had been tested using this method, they would have gotten a diagnosis of Lyme disease. So it was able to pick up, the Lyme-Seq assay was able to pick up those positives in those groups of patients who would be negative, most likely negative, on the serological assays. So that was a very large positive finding as well. So this is the TGen's initial findings and their analysis that I just was able to borrow the slide from. So the assay clearly separates Borrelia species. It clearly identifies them. It identifies other tick-borne pathogens as well, and they found that both in ticks and they found that in clinical samples. It detects targets down to less than 1 CFU, which actually stands for colony-forming unit, and just think of it as a cell, a bacterial cell. So they can detect the presence of Borrelia and other things at a number of less than 1 cell per sample, okay? And then the version 2, I forgot what version 2 did, but it was something good, so just remember that, okay? So applications, limitations, future directions. So the diagnostic accuracy of Lyme-Seq is equal to PCR in the subgroup of patients that are, the bullseye rash followed by positive serology. So in other words, those in the confirmed group. So it is as good as, and then better than PCR and other, also for the serology group, for those groups of patients that are the problem, all right? So the bullseye rash people are not the problem. It's the chronic persistent symptom group that is a problem when it comes to diagnosis and obviously treatment. So, but what that also says is that it's clear that using methods to detect nucleic acids, and this is what Garth just said too, it remains problematic because of the low numbers of bacteria. Actually, nobody knows why it's problematic. It just is. It's difficult to isolate nucleic acids to an extent that they are able to be amplified or they're actually able to be sequenced or anything else. So why is that? And we've alluded to before that these are remarkable bacteria. They are remarkable. They have rewritten the textbooks on microbiology, without a doubt. And so what is the significance of those non-spirochetal forms when it comes to this kind of assay? None of that has been investigated scientifically, and it most certainly needs to be as we're working our way into coming up with better diagnostics. There are technological limitations that have to do with just that process alone. Blood may not be the best specimen to use. Although it would be great if it was, because from a physician perspective, and I look at the physicians in the group to say this, what they want to do is to be able to draw blood and send it to a lab and get results. So as opposed to other more invasive methods or anything else, it would still be the best way to get an accurate diagnosis. So there needs to be a way to do something about that. And so next steps in this, it would appear we have an excellent framework to move forward with, and what we need to do is to come up with a way to induce those bacteria, to find out what is going on with those persistent with the persister type cells, with the round bodies, whatever you want to call them, because there's like 16 names for these non-spirochetal forms of Borrelia. And now, even the spirochetal persisters, what is going on with them, so that we can find out maybe the targets that we're using to detect their nucleic acids need improvement too. So that is the step forward, moving on from that. But at the moment, I'm personally very happy to report that what has been developed up to this stage does appear to be, it is certainly going to increase the detection ability in those patients that have issues getting blood, getting positive test results for that. So I also just want to say, on the behalf of Focus on Lyme, thank you to the TGen team. And can you change this just one last time? Thank you. So to the translational genomics team who's been working on this, headed up with Dave Engelthaler and also Jolene Bowers and Elizabeth Dreby, who are actually the ones doing the hard work of making it into something that is usable. At Northern Arizona University, of course, Paul Keim, who has provided his insight on microbiology all the way through this, and also Nate Nieto, who has worked with that team extensively, provided the ticks, also did backup testing on PCR. These are the people that have been working very hard to try to make that happen. I also want to say thanks to the BHH Legacy Foundation, the BHHS Legacy Foundation, for providing funding in addition to Focus on Lyme for moving this forward to the point that it has. So in 2 years, we went from nothing to an assay that it looks like there's some promise there. So thank you, and I think I'm standing in the way of some tequila and lime, so.
Thank you, Holly.
Yeah.
And